Welcome to Ask Prep, your trusted companion for NEET and JEE preparation. Today we will study about a very important topic of biology, which is plant tissue culture, micropropagation, somatic embryogenesis. These terms might sound technical, but by the end of this session, you will understand not only what they mean but also why they are so important, both for your exams and for real-world applications in agriculture, horticulture, and biotechnology. Let's begin with the idea of plant tissue culture. Imagine if you could grow a whole plant from just a small piece of tissue, like a leaf, stem, or even a single cell. That's exactly what plant tissue culture allows us to do. It is a technique in which plant cells, tissues, or organs are grown in an artificial medium under sterile and controlled conditions. The fascinating thing about plant cells is that they have a property called totipotency. This means that each living plant cell has the potential to regenerate into a complete, whole plant under the right conditions. This unique ability makes tissue culture possible and opens up incredible opportunities for large-scale plant production. Now, why is plant tissue culture so important? For one, it allows us to produce a large number of genetically identical plants, called clones, in a very short time. This is especially useful when we want to multiply plants that have desirable traits like high yield, disease resistance, or ornamental value. Tissue culture also helps in conserving rare or endangered plant species, producing disease-free plants, and enabling advanced research in plant breeding and genetic engineering. One of the most widely used applications of plant tissue culture is micropropagation. Micropropagation refers to the rapid multiplication of plants using small tissue pieces called explants, which are grown in nutrient media under controlled laboratory conditions. The process typically involves several stages, starting with the selection and preparation of the explant. The explant is usually a small section of the plant, like a shoot tip, leaf, or node, which is carefully sterilized to remove any microbial contamination. This is important because even a tiny bacterial or fungal infection can destroy the entire culture. Once sterilized, the explant is placed onto a culture medium that contains essential nutrients, vitamins, plant growth regulators, and sometimes sugar as a source of energy. Depending on the balance of hormones in the medium, the explant may start producing multiple shoots, which can then be separated and further cultured to increase the number of plants. Eventually, the small plantlets are transferred to a rooting medium, where they develop roots, and finally, they are moved to soil or potting mix for hardening before they are ready for field planting. Micropropagation has many advantages. It allows for the rapid and continuous production of high-quality, uniform plants regardless of the season or location. It is especially useful for plants that are difficult to propagate through seeds or cuttings, such as bananas, orchids, or certain ornamental plants. Micropropagation also helps in producing disease-free planting material, especially important for crops affected by viral or bacterial diseases. However, it is important to manage the process carefully because maintaining genetic stability is crucial. Any unintended genetic variation, called somaclonal variation, can lead to undesirable traits in the propagated plants. Another fascinating outcome of plant tissue culture is somatic embryogenesis. Somatic embryogenesis is the process by which somatic, or non-reproductive, plant cells develop into structures that resemble embryos, eventually giving rise to whole plants. Unlike sexual embryos, which come from the fusion of gametes during fertilization, somatic embryos develop from ordinary body cells like those from leaves or stems. These somatic embryos can pass through the same developmental stages as a zygotic embryo, forming a shoot, root, and eventually an entire plant. The process of somatic embryogenesis generally begins with the induction phase, where the somatic cells are stimulated, often by stress or specific growth regulators, to become embryogenic. Once the embryogenic cells are formed, they develop into embryonic structures in a series of stages: globular, heart-shaped, torpedo-shaped, and cotyledonary stages, much like a zygotic embryo. These somatic embryos can then be matured, germinated, and converted into plantlets, which are ready for planting. Somatic embryogenesis offers several exciting applications. It enables the large-scale production of plants, just like micropropagation, but with some added benefits. Somatic embryos can be stored for long periods, encapsulated to create artificial seeds, or even used in genetic engineering and cryopreservation. This technique is widely used in the forestry industry, horticulture, and in the conservation of endangered plant species. The importance of tissue culture techniques, including micropropagation and somatic embryogenesis, extends beyond just multiplying plants. They play a crucial role in biotechnology research, allowing scientists to study plant development, introduce new traits, and conserve valuable genetic resources. For example, tissue culture is essential in producing transgenic plants, where new genes are introduced to improve crop performance, or in developing virus-free planting material for crops like potatoes, sugarcane, and strawberries. It is also worth noting that these techniques require careful control of environmental factors such as temperature, light, humidity, and the composition of the culture medium. This is why tissue culture labs are equipped with sophisticated equipment like laminar airflow cabinets, autoclaves, and growth chambers. Sterility is absolutely critical at every stage to prevent contamination, and researchers working in tissue culture need to follow meticulous protocols to ensure success. To summarize, plant tissue culture harnesses the power of totipotency, enabling scientists and farmers to produce large numbers of high-quality, genetically uniform plants through methods like micropropagation and somatic embryogenesis. These techniques help increase agricultural productivity, conserve plant biodiversity, and support research and innovation in plant science. They represent the intersection of fundamental biology with cutting-edge technology, demonstrating how scientific knowledge can be applied to meet global challenges. Thank you so much for being a part of the Ask Prep community. We truly appreciate your time and dedication to learning, especially when it comes to mastering biology, a subject that's not only essential for exams like NEET and JEE. If you found this video helpful, please give it a thumbs up, share it with your friends who are also preparing, and make sure to subscribe to our channel so you never miss an update.